A very good afternoon. So, like last week, we started with the Drosophila development. We started with the Drosophila axis formation. So, we'll be continuing with that, and we'll be studying about Drosophila axis formation in this class as well. And we'll try to solve questions regarding Drosophila axis development in this class. Okay. Yeah, my voice is not clear. Can you hear me now? Can you hear me, Shubhoy? Okay, just a minute. Now, can you hear me? Is it coming now or is it not coming still? There's the problem. Not clear. Okay, I think okay, it's coming. It's still not clear. Can you hear me? Is my voice audible now? Okay, fine. Now we'll start the class. So last week we started with Drosophila axis development and we were left with one question. So we first solve that question and then we move to this week's questions. So before starting, let us quickly revise what we have learned in the last class. Okay. So we were talking about the different genes, different types of genes which are involved in Drosophila axis development. Okay. So So when we talk about Drosophila axis development, we started that there are two types of genes. First are the maternal genes and the second ones are the zygotic genes. Okay. Maternal genes. And second one are the zygotic genes. So can anybody tell me the examples of maternal genes? Any one or two examples of maternal genes? So how much in this? Okay, this I have to check. Anybody knows any example of maternal gene? So is bicoid a maternal gene or not? Tell me. Hmm? Is bicoid a maternal gene or not? Tell me. Yes, bicoid is a maternal gene. And then what else? What all are other maternal genes? Bicoid, nanos. Bicoid is a maternal gene. Nanos is a maternal gene. You have to mix it something. These are all maternal genes. And what are the different types of zygotic genes? Yes. So bicoid, nanos, caudal, hunchback, these are maternal genes. And hunchback also comes under the category of zygotic genes that we'll deal later. And then what are the zygotic genes? What are the different types of zygotic? Hmm? So there are different types of zygotic genes such as gap genes, pair rule genes, segment polarity genes, and homeotic selector genes. This is what we have started with in the last week class. And you can see that first you have maternal genes. So maternal genes are the genes which are expressed in oocyte before the fertilization. So before the fertilization has taken place, these maternal genes are expressed in the oocytes. So these bicoid, it is expressed in the oocyte before the fertilization. So at what place does bicoid is expressed? It is expressed from the, secreted from what? It is secreted from the nerve cells. So one important point to note over here is, this is anterior and this is posterior. So bicoid is initially synthesized from the posterior end, and nanose is initially synthesized from the anterior end. Okay, I'm talking about the synthesis. So now bicoid is initially synthesized from the anterior end, but it might, sorry, posterior end, but it migrates towards the anterior side. Likewise, nanose is initially synthesized from the anterior end, but it migrates towards the posterior side. So it is an important point to note that although nanose and bicoid are synthesized from different ends, but eventually they migrate, and bicoid is responsible for anterior side axis formation and nanose is responsible for the posterior axis formation. So very quickly, if we see here, this is the diagram that I wanted to explain before starting the questions. Okay. So are you able to follow me? If there is any problem regarding the voice or any other, you can just message in the message box or type in the message box. Okay. Fine. Take it. See here. This is the oocyte mRNA before fertilization. So this is the anterior axis and this is the posterior axis, and this y-axis represents the concentration. So here you can see that bicoid mRNA. Okay, so before fertilization, the maternal genes are present as mRNA only. They are not translated into protein. The translation of these mRNA into proteins takes place only after the fertilization has taken place. So initially, before the fertilization in the oocyte, so in the anterior end, your bicoid mRNA is present, and at the posterior end, your nanose mRNA is present. And the levels of hunchback and caudal are same. They are same throughout the axis. That means throughout the anterior and posterior axis, the expression levels or the concentration of hunchback and caudal mRNA are same. As soon as fertilization takes place and there is early cleavage, then what happens? You can see here. Then what happens? You see that now this bicoid is converted into protein, it is translated into protein. So the translation of all these mRNAs, all these maternal genes, the translation of all these maternal genes takes place only after the fertilization has taken place. So after the fertilization, you can see now this bicoid, it acts as a morphogen, it forms a gradient, and the place where this bicoid is present in the greatest concentration, that area forms the anterior region. And the place where this nanose protein is present at greatest concentration, this area forms the posterior region. And you can see that the level of hunchback, it is also eventually decreasing from anterior to posterior. Likewise, the concentration of caudal is also increasing, is showing a gradual decrease from posterior to anterior end. So if we want to study this in a flowchart form, if we want to study this in a flowchart form, you can see that bicoid mRNA, after the fertilization, it is converted into bicoid protein. That means bicoid mRNA is translated into bicoid protein with the help of some other factors, which are these strophin, cortex, and everything. Now, this bicoid protein inhibits the translation of caudal mRNA, which is present in the anterior side. Okay, so in the anterior axis, although caudal and see before fertilization, Here you can see that before fertilization, this hunchback and caudal mRNA is present throughout the anterior to posterior axis. But after the fertilization has taken place, this bicoid mRNA it represses or inhibits the translation of caudal mRNA to caudal protein in the anterior end. So this caudal also mRNA was present in the anterior end, but there is no protein in the anterior end. Okay. Likewise, in the posterior end, you see this nanos mRNA is converted into nanos protein. Now, although hunchback is also present in the posterior end, hunchback mRNA is also present in the posterior end, but this hunchback mRNA cannot be converted into hunchback protein. Why? Because nanos inhibits this translation. So, in this way, although hunchback and caudal mRNA were present throughout the axis, but hunchback protein is specifically expressed in the anterior region and caudal protein is specifically expressed in the posterior region. So, this becomes an important point for you. Is it clear to everybody? Is it clear? If there is any question, you can ask. Okay. So, shall we move to the questions now? So I just wanted you to revise this to recapitulate it because it's important. And also we have studied in the last class about gap genes, pair rule genes, segment polarity genes, and homeotic selector genes. And we'll try to study more about these genes eventually as we move through our class. So first we'll start with a question that we left in the last class because of time constraint. We couldn't complete that question. Okay. So this is the question. The question says, during embryogenesis, the protein levels of early Drosophila embryos were recorded. So these are the protein levels. Based on your understanding of regulation of anterior posterior axis formation, choose the correct statement. What are the statements? Normally, hunchback expression is both maternal and zygotic. In condition A, hunchback can be an inhibitor of anterior expression of bicoid. Caudal expression will be lower in condition B, and embryos in graph B may have had overexpression of strophin. Embryos in graph B may have had an overexpression of nanos, and graph B could be a result of overexpression of humilio expression. So these are your options. I'll give you 3 minutes. Try to solve this question. After 3 minutes, I'll switch the screen. Okay? Now try to solve it. After 3 minutes, I'll switch the screen. 2 minutes left. So you need to correlate the options with the expression or the fold levels of the two proteins, bicoid and hunchback. Okay. So see what is the difference in the level of bicoid and hunchback in graph A and graph B. And this is the fold activation. That means their expression level we're talking about. 1 more minute left. Okay, now I'm switching the screen. Let us see what you have answered. Hmm. Okay, so Ajay has answered A, C, E, and F. And what about the others? Why others have not tried to answer the question? Hmm? Chalo, let's see the options. A, C, E, and F. Let me write it. Hmm. Okay, so let us try to solve this question. So the question says, during embryogenesis, the protein levels of early Drosophila embryos were recorded. Based on your understanding of regulation of anterior posterior axis formation, choose the correct statements. So in graph A, you can see that the expression of bicoid, the levels of bicoid is lesser than that of hunchback. So here hunchback is more. Here bicoid is more than hunchback. Okay. Now let us try to see the options. In normally, hunchback expression is both maternal and zygotic. Is it true or false? It is true. Hunchback can be regarded both as a maternal gene and a zygotic gene. Only um, mm, why we can say this? Because mRNA is there in the maternal, but the real expression of hunchback comes only when uh, uh, it is after the fertilization has taken place and it is translated. And it is hunchback can be taken as a gap gene as well as a maternal gene. So that is why all the gap genes are zygotic gene except for hunchback, because since although hunchback is also a gap gene, but it can also come under the category of maternal gene because it is present before the fertilization has taken place. Although its level is same, but it is there. Then in condition B, hunchback can be an inhibitor of anterior expression of bicoid. Can it be true? No, because bicoid promotes the expression of hunchback. Bicoid, it promotes the expression of hunchback. And therefore, both bicoid and hunchback are expressed in the anterior. So, it cannot be the case that hunchback inhibits bicoid. No. So, this is false. Then, next is caudal expression will be lower in condition B. See, in condition B, you have bicoid is expressed more. Now, we know that bicoid will inhibit caudal. Therefore, caudal expression will be lower here because bicoid is expressed greatly here. So, this is true. Then, next is embryos in graph B may have an overexpression of stophin. So is it true or false? It is false. Why? Because this stophin is involved in the localization of bicoid mRNA. So although stophin mutation affect bicoid distribution, but here they are saying that this statement does not fit to the expression profile in graph B, because overexpression of strophin, it will affect bicoid localization more than the fold change. Okay, so therefore this statement again becomes false. Then what is the next? Next is embryos in graph B may have an overexpression of nanos. So here, can there be an expression of nanos? There can be an expression of nanos. Why? Because if bicoid is expressed more, that means it is talking. We are talking about the interior side, and hunchback is also there. So there is nothing which can be said that nanos would be inhibited. So this statement becomes true. And then F is graph B could be a result of overexpression of Pumilio expression. So What is the function of Pumilio? Basically, it works with nanos to repress the hunchback translation in the posterior side. So you should understand that although hunchback mRNA is present in the posterior end, but hunchback protein is not formed in the posterior side. Why? Because nanos, it represses the translation of hunchback proteins. To do so, nanos takes the help of Pumilio. So Pumilio, along with nanos, it tries to repress the hunchback translation in the posterior end. So if Pumilio is overexpressed, what does it mean? It means that hunchback would be repressed in the posterior region. Yes, but it is not clear why would this led to the pattern B, like this graph, where hunchback is still present, although it is less sharp than A. So now this option becomes a little confusing. Why? Because you can see here it is approximately, yeah, it is approximately 1.7 to some fold expression, which is still a good expression only, but compared to this, it is very less. So whether this much expression can be considered a decent level of expression or not, so there has to be a threshold which should be given according to me, because if there is no threshold given, then we cannot say that hunchback is completely repressed or not. In the answers, yeah, graph B could be a result of overexpression of Pumilio expression. Now here they are saying, so according to this, F is correct. That means we cannot consider this much expression as a standard expression. Could you understand what I'm trying to say? Could you follow what I'm trying to say here? See, if you look at the expression level of uh, hunchback, comparatively, in comparison to bicoid, it is less. So in that way, this becomes correct. Why? Because if Pumilio is overexpressed, it will inhibit the hunchback translation. Okay, because here what is written, option F. Try to understand here that graph B could be a result of overexpression of Pumilio expression. Why they are saying that graph B could be a result of overexpression of Pumilio expression? Because hunchback is getting repressed. Now, for this, to say whether hunchback is getting repressed or not, you need to have a control. In control of which you can say that okay, earlier hunchback was expressed this much, and now because Pumilio is overexpressed, so there is a control in comparison to it has down regulated. But here there is no control, so it becomes a little difficult to say whether this much expression can be considered overexpression or underexpression. But we can compare with here, it's not a control, these are two test conditions only, but in comparison to graph A, in graph B, hunchback has repressed. So in that accordance, option F also becomes correct. Otherwise, there is little disparity. Okay, I hope you could follow with the question. I hope you could follow with what I'm trying to say. See, always understand, leave this question here. I think you have to consider these two values only. But whenever you do any kind of comparative analysis from the word itself means that you have to compare. So if there is one graph, it is showing expression level of 4, let's say. So 4-fold is a good expression. And but if the control shows the expression level as 10, then in comparison to this control, it is underexpressed. If you have another control which shows some expression level of, let's say, 2, then according to this is overexpressed. So that is why we call it as comparative analysis. It all depends upon how much expression our control does. Okay, so if you have done real time experiments, so to avoid this, we normalize. Okay, so to avoid this kind of confusions, we normalize. So we normalize with respect to a positive control so that our control always shows one fold. If anything is above one fold, we consider as a positive regulation. If anything is below one fold, we consider it as a down regulation. Okay, but that is a different story. I just want to tell you that whenever you're doing a comparative analysis, you always have to have control. Whenever you're doing experiments, also there also you have to have a control. Anyways, now let us go to the next question. Yeah, easy one. The question says, which of the following are the characteristics of pair rule genes? They are expressed in vertical stripes along the embryo. They are expressed in horizontal stripes along the embryo. Their expression begin after cellularization. And the last one is modular enhancer play a crucial role in determining their expression patterns. So, what are the characteristics? 2 minutes to solve this question. It's easy one. Try to solve, then I'll switch the screen. One more minute left. Okay, I'm switching the screen now. Hmm. So, nobody has answered. Okay, Ajay has answered A and D. Others? Shubhamoy, Aditya, what do you think should be the answer? It's an easy one. You should be able to try at least. So just, okay, Shubhamoy has answered D. Good. Aditya, no? At least try to attempt. Just say randomly any question. No? Okay, fine. See, question says, which of the following are the characteristics of pair rule genes? So, um, just a minute. So, what are pair rule genes, first of all? So, these are the zygotic genes, and they establish uh, segments like if you know this is if this is your embryo, okay, suppose. So, this like this, they try to establish the segmentation. Approximately 7 segments are there. So, they divide the embryo. So, these pair rule genes, they divide the embryo into seven stripes. Okay. And these stripes basically represent a segment. So this is the embryo. Along the length of the the um, embryo is getting divided into segments. So first option would be correct. That means they are expressed in vertical strips along the embryo. So A is correct. They are not expressed in horizontal strips. Why? Because along the length of the embryo, this is your embryo, and this is the length. So along the length of the embryo, your pair rule genes divide. So if they divide like this, then only along the length they will be able to divide. Okay, so they will divide. They are expressed in vertical strips. They are expressed in horizontal strips. No, their expression begins after cellularization. No. So now, what is cellularization? Can anybody tell me what is the meaning of cellularization? Anybody? Okay. First of all, this option D, modular enhancements play a crucial role in determining their expression pattern. True. So your correct options are A and D. Let us see. Yes. Now, can anybody tell me what is the meaning of cellularization? Anybody who knows what is cellularization and why it is important to study in context of embryo, specifically Drosophila embryo development? No? Okay. I'll try to explain as simply as I can. So, always my focus is that I try to explain the concept which comes behind that particular question. I always say don't focus on the quantity, focus on the quality. That means don't focus how many questions we are able to solve. Focus whether you are able to solve 6 or 7 questions, but are you able to understand the concept? So these or whatever questions that we are solving, be it 6 questions, 10 questions, 10, 15 questions, the idea should be that that particular topic should be clear, and that particular topic should be overall covered with in those question framings. Anyways, so now we'll see what is cellularization. So in Drosophila development, what happens is initially there is the formation of syncytial blastoderm. What is the meaning of syncytial blastoderm? It means that syncytium forms. What does it mean? It means that there is only nuclear division and no cellular division. That means the nucleus keeps on dividing within the blastocyst, within the blastoderm, but the cellular division does not take place. So if this is your embryo, your nucleus is getting divided, but membrane around the nucleus is not formed. So it is a syncytium. Then what happens that approximately during the 10th cleavage cycle, okay, during, I'll just approximately during the 10th cleavage cycle, what happens is that your nuclei, it reaches the periphery. Okay, nuclei, it reaches the periphery and it forms a cellular blastoderm. So if this is your embryo, let's say, and these are your nuclei. Okay, so earlier what was there? Nuclei was dispersed throughout the embryo, and now they reach the periphery, and after reaching the periphery, what happens that now cellular membrane around the nucleus forms. Can you understand like this? Just if there is only one nucleus, I'm just trying to explain you what happens. So this process is known as cellularization, and this is a cellular blastoderm. And a blasto and this one where the nuclei are present, but the nuclear membrane around the nucleus is not formed. That is a syncytial blastoderm. So, is this clear to everybody? The difference between the syncytial blastoderm and a cellular blastoderm. Is it clear? Shubhamoy, Ajay, Aditya, is it clear to everybody? I hope it is clear. Now I'll move. Okay, if there's any problem, you can let us know. Okay, next question is identify the ABC in the following figure. So this is a mechanism which we will be studying just after this question. You have to identify what A, B, A, B, C, D signifies. So whether A is wingless, B is hedgehog, C is the transcription of armadillo, D is the transcription of engrailed and hedgehog. Whether A is hedgehog, B is wingless, C represents the transcription of wingless, D is the transcription of engrailed and hedgehog. Whether A is hedgehog, B is wingless, C is the transcription of engrailed and hedgehog, D transcription of wingless, and the last option is A is wingless, B is hedgehog, C is transcription of hedgehog, and D is the transcription of engrailed. So this is a mechanism. You have to tell that what A, B, C, D refers to. So I'll give you 2 minutes to solve this question. After 2 minutes, I'll switch the screen. Okay? It's very easy. Pay attention. If you know how this cycle works, okay, then it is easier for you. It's 4:42. At 4:44, I'll switch the screen. Okay? One more minute left. One more minute left to solve the question. Okay, now I'm switching the screen. See, okay. So Ajay has answered. Okay, Aditya has also answered B. Okay, good. Ajay has answered B. Anybody else who wants to try? Huh? No? Okay, fine. So the correct answer is A is hedgehog, B is wingless, C is transcription of wingless, and D is transcription of engrailed and hedgehog. That means B is the correct answer. Now we'll see how it goes. B is the correct answer. So this is the actual um this thing mechanism. So let us start from here. Okay, I'm going to explain this in detail. So just keep attention, pay attention here, and try to listen to the story. So what happens? These are the wingless producing cells. So what are wingless and what are these are what genes? Wingless and engrailed. Under which category does they come? Hmm? So basically, we are talking about segment polarity genes. Okay, segment polarity genes. So after this question, I'll just in a brief explain you about the different genes. That means the gap genes and pair rule, segment polarity, and all that, because it is very, these are the basics for Drosophila axis development. Okay, so this is a wingless producing cell. This is a wingless producing cell, and this is a engrailed producing cell. This is engrailed producing cell. Now, what happens in the wingless producing cell? Transcription of the wingless protein takes place. Wingless protein is a secretory molecule. So, after the transcription and translation of wingless, this wingless it is secreted out of the cell and it goes to the nearby cell, which is the engrailed producing cell, and it binds to the engrailed producing cell with the help of this frizzled receptor. So, with the help of this frizzled receptor, this wingless protein goes and binds to it. Then the downstream processing takes place and there is transcription of engrailed and hedgehog both. So, this is an engrailed producing cell and it also produces hedgehog. So, when this wingless comes and binds, there is transcription of hedgehog. And this hedgehog is also a secretory molecule. So after its production, it is secreted and it again goes and binds to the this wingless producing cell through PAST receptor. So hedgehog is binding to the PAST receptor, and then again which leads to the transcription of wingless. So it's like a cycle. So in this way, the transcription and translation of the segment polarity genes is controlled. Is this clear to everybody or not? So, just you have to understand that there are two cells nearby. One is your wingless producing cell, other one is your engrailed producing cell. Your wingless producing cell transcribes wingless and wingless is produced. Since this wingless protein is a secretory protein, it is secreted out of the cell. It goes and binds to the frizzled receptor which is present in the nearby cell, which is an engrailed producing cell. Then, through all the downstream processing, transcription of engrailed and hedgehog take place. This hedgehog is secreted, it binds to the PAST receptor, and ultimately, which leads to the transcription of wingless. Is it clear? Is it clear or not? Is this pathway clear to everybody? Yes or no? At least say something. No, no response, huh? So either everyone is sleeping or they understood everything. Anyways, so since there is, yeah, okay. So Aditya and Shubha has replied. Good. So now can you tell me that this pathway is an example of which, I mean, this mechanism is an example of which pathway? Can anybody tell me? This mechanism is an example of which pathway? So initially, in the initial, uh, okay, so Aditya is saying Artika pathway. Anybody else who wanted to give a try? So if you remember in the initial classes, we discussed that there are four major pathways which are involved in this development of Drosophila. Okay, so one was Artica pathway. What were the other pathways? Can you guess? We studied about wind pathway. Could anybody recall wind pathway? What happens in wind pathway? Can anybody tell me? Huh? What happens in wind pathway? In the, yeah, okay, hedgehog wind paracrines and lilies. Okay. So what happens in the wind pathway? Can anybody tell me? No. So in the normal wind pathway mechanism, just a minute. What happens? Yes. So if we try to read about the canonical, so every pathway can have a, like in developmental biology, these pathways have a canonical version and the non-canonical versions. So canonical versions are the ones which are the general and the most popular and the most used pathways in the cell, and non-canonical are the exceptional pathways which are used at some or more places but not commonly. So canonical pathways are the commonly used pathways in the cell. So in the canonical pathway of Wnt protein, sorry, in the canonical Wnt pathway, Wnt is a short form of wingless. So Wnt binds to the Frizzled receptor, and then when Wnt binds to the Frizzled receptor, there is a beta catenin destruction complex. This beta catenin destruction complex cannot destroy the beta catenin, and therefore the transcription takes place. And when Wnt protein is not present, what happens? Then this beta catenin destruction complex, which consists of disheveled GSK3 beta and some other proteins, they destroy the beta catenin through the process of ubiquitination, and therefore transcription does not take place. So this is an example of a Wnt pathway where the ligand, which is a protein, it binds to the Frizzled receptor, and the destruction complex, which actually destroys the beta catenin, is not able to bind to the beta catenin. Okay, and beta catenin here is armadillo. So armadillo is nothing but beta catenin here. So this is an example of Wnt pathway, and this is hedgehog pathway. Is it clear to everybody? I hope it is clear. If not, you should go and revise the pathways. So we have already discussed in detail about the pathways. So there we have just discussed the general mechanism of how a pathway works. But now here comes the application of those pathways in various signaling in the developmental process of Drosophila. Okay. Okay. Now we move to the next question. Yeah. Easy one. Antennapedia complex contains what? Labial, and, antennapedia, sex form reduced, deformed, Proboscipedia, UBX, ABADA, ABDB, labial antennapedium deformed, sexcom reduced, deformed, or probospedia. So I will give you 2 minutes to solve this question. Okay, try to solve this question. After this question, we'll try to just quickly study about the different kinds of genes. I mean, the different types of genes, zygotic genes which are involved. Okay. 1 more minute left. Okay. So Ajay has answered A and Aditya has also answered A. Okay, and what is A? Yes. So A is the correct answer. So the correct answer is hmm, the correct answer is labial, antennapedia, sexcom reduced, deformed, and probospedia. So basically, what is antennapedia? Can anybody tell me what is antennapedia? These are the examples of these are the examples of homeotic genes. So if you see here, so if you talk about homeotic genes, so there are generally two major complexes which comes under homeotic genes. And what are those complexes? We are talking about homeotic genes. So the first complex is antennapedia complex, and second one is the bithorax complex. You can see here antennapedia complex and bithorax complex. Now, one interesting thing about the homeotic genes is that you see from left to right. Okay, this is the leftmost part and this is the rightmost part. Now, left to right arrangement of these genes, it corresponds to the anterior to posterior axis formation. What I'm saying? I'm saying that left to right arrangement of these genes, it corresponds to the anterior to posterior axis formation. See here, this gene, they are responsible for the formation of head. Then deformed proboscidea, this particular area, plus lab, that means labial. These all are responsible for the head formation. Then sexcom reduced and antennapedia. This particular region genes are responsible for your thorax formation. Then you can see ultrabithorax. This is ultrabithorax, abdomen A and abdomen B. These genes are responsible for the abdomen formation. Okay, and you can see here also, see, just go with the color coding. This is the head region. This is the head region. These are the thorax region, T1, T2, T3, and this is the abdominal region. So see, head region is specified by labial and deformed and proboscidea. You see here, this is the head region. Then your thorax is specified by sexcom reducer and antennapedia. Then T3 segment means the thorax 3 segment is specified by UBX, that means ultrabithorax. And then this abdominal, different segments of abdomen A1, A2, A3 till A4, it is specified by abdomen A. And then abdomen B, mostly for the last segments of the abdomen. So this means that the segments that has already been formed, the significance to those segments, the identity to those segments are given by your homeotic selected genes. And these homeotic selected genes are nothing but the interaction between gap and pair rule genes. They regulate the expression of homeotic selected genes. Okay, so the interaction between your gap genes and your pair rule genes, it decides what kind of expression pattern does homeotic selected genes will have. So now we'll try to very quickly we'll try to summarize all the different types of zygotic genes here. Is this question clear to everybody, or is there any confusion? Is this concept clear? The homeotic selected gene concept. Important thing to understand here is that left to right arrangement of these homeotic selected genes, it corresponds to the anterior posterior axis formation. This is important to understand here. Okay. Now, just a minute. Hmm. So, one thing that we know about the Drosophila is that it has a segmented body. Okay. Even our body is also segmented. Segment means that we have a head region and abdominal region. So, Drosophila, the segmented body, this is regulated through a hierarchical gene regulatory network. And the four main categories of genes that control this process are the gap genes, pair rule genes, segment polarity genes, and homeotic selective genes. And each play a specific role in defining the body's segment from head to the tail, that is, from the anterior to the posterior axis. So, if we see this particular uh, graph, it is here, I think, to see where it is. No, it's in first picture. So from here we'll start with, see the hierarchy. First there are maternal genes, then there are grab genes, then there are pair rule genes, then you have segment polarity genes, and then it comes to the homeotic selector genes. So gap genes basically it define large and continuous segments of the embryo, which provides broad positional information along the anterior posterior axis. So these genes are expressed in broad domains. You can see these are very broad domains, and mutation in gap genes leads to large loss of large body regions. Therefore, there will be gap in the gene in the body. So therefore, they are known as gap genes. So all the name of the genes in your developmental biology, they are named according to the morphology which happens when this that particular gene is mutated. So if there is a mutation in the gap gene, there would be gaps. Therefore, this particular gene is named as gap gene. So gap genes they specify the broad domains. Okay, the large and continuous segments. And what are the examples of gap genes? We have hunchback, we have cripple, we have giant, okay, then we have NRIPS, and which each and every gene has a different role. Then comes the pair rule genes. Now, basically, they form alternating pattern of alternating segments, which are known as para segments. And they divide the embryo into 7 stripes, and each have a particular function. So, pair rule genes are expressed in stripe that span every other segment along the length of the embryo. And what are the examples of pair rule genes? They are even skipped, Fushi Tarazu, Brunt. They are examples of gap genes, sorry, pair rule genes. Now, pair rule genes, see, understand, gap genes regulate the expression of the downstream genes. So what are the next downstream gene of gap gene? Pair rule genes. So in the immediate manner, gap genes regulate the expression of pair rule genes. Pair rule genes regulate the expression of segment polarity genes, and segment polarity genes regulate the expression of homeotic selector genes. So pair rule genes, it will regulate segment polarity genes. Therefore, segments are further refined. So there are broad segments. Those broad segments were further refined. Then even more refinement will take place, and after that, purpose of each segment would be given. So this is a hierarchy in which it is going. So we are dividing the whole embryo into segments, and we are refining it. So after each hierarchy, the segments are getting refined. Here they were very broad. Now we got 7 segments. It will be even segmented by the segment polarity genes, and then comes your homeotic, which will give function to each segment. Okay, so now comes your segment polarity genes. So basically, segment polarity genes define the anterior posterior polarity within each segment and ensure proper boundary formation between them. So within each segment also now there would be anterior posterior polarity given by the segment polarity genes. And basically it gives it leads to even finer subdivisions. There were divisions, even finer subdivisions. And what are the examples of your segment polarity genes? These are wingless, engrailed, hedgehog. These are the examples of segment polarity genes. So, segment polarity genes are basically involved in the communication between cells to ensure proper segment polarity and segment formation and polarity. So, within each segment, proper polarity formation takes place with the help of segment polarity genes. And then come your homeotic genes. What are homeotic genes? So basically, now we have made the segments. Up till here, the segment process is completed. Making of segment process is completed. First, there were broad segments. Then we have further segments. Then we have even refined, we have given polarity to the each segment also. Now, when everything is refined, next comes giving identity to each segment. So these homeotic genes, they gives identity to each segment, and it ensures that a correct structure is formed at the appropriate location. Okay, it is important that correct structure forms at the appropriate location. That is why when we were dealing here, we told that the expression of these genes, where they are expressed, it will signify where head will be formed, where thorax would be formed, and where abdomen would be formed. And if there is any mutation in the homeotic genes, it can lead to homeotic transformation. That means legs can grow in the place of antenna. So there can be complete malformation of the body structures. So overall, if we want to study, we can say that gap genes give us a broad regional definition. Pair rule genes divide the embryo into alternating segments. Segment polarity gene, it defines segment boundaries and polarity within the segments. And homeotic genes, it give identity to each segment, whether that segment will signifies your head or thorax or the abdomen. So I think I tried to explain all the uh, different kind of polarity genes. Again, now we'll go to the next question. Okay. Hmm. Okay. Homeotic protein bind to DNA via their homodomain, homeodomain, UBX or HRE region. 2 minutes. After 2 minutes, I will switch the screen. It's 5:11. I'll switch at 5:30. 1 more minute left. It's an easy one. You should be able to answer it quickly. Okay. So, what you have answered? So, Ajay has answered B. Okay. What is the B? Okay. And anybody else? Shubhamoy, Aditya, what do you think should be the answer? Hmm? Okay, everybody thinks B and B is the correct answer. So basically, homeotic genes, they contain a homeodomain, and this homeodomain has 60 amino acids that are conserved among various homeotic genes, and they basically bind to DNA because and their transcription factor. So this homeodomain is basically transcription factor. That means they are proteins which consist of 60 amino acids, and this is coded by a region of DNA which is approximately 180 base pairs, which is known as homeo box. So this homeo domain is coded by a 180 base pair region of DNA, which is known as homeo box. Okay, so now can anybody tell me what is the difference between a homo domain and a homeo domain? This, can you tell me what is the difference between homo domain and a homeo domain? Hmm? Anybody? Homo is similar. Okay, then fine. Homo is similar. Then so what? Homeo means maintain. So what inference can you get from here? What is the difference between a homo domain and a homeo domain? Hmm? Just an E? So homeo domain is a region of a protein. Okay? Homeo domain is a region of a protein. So when we say homeo domain, when we say homeo domain, we are actually talking about a region of a protein. And here this region works as a transcription factor. But when we talk about homodomain, it is a term which is mostly used when we want to specify a particular region of a gene. That means on a gene level. So homeodomain term we use on a protein level, which is a region of a gene, whereas region of a protein, whereas homodomain is a region of a gene. I'm just writing. When it is homo, it specifies a region of a gene. When it is homeo, it specifies a region of a protein. This is the most basic way in which I can explain you. Okay, fine. Now, next question is, hmm, given below are the two graphs depicting gene expression in the anterior most area of Drosophila embryo. Identify the correct inferences based on your understanding. First is, embryo A may have been bicoid deficient. B, embryo A may have hunchback deficient. C, embryo A may have had an overexpression of nanos in the thoracic region. D, trithorax binding to lab and DFD regulatory regions of embryo A caused the expression pattern. E, embryo B may have had ectopic expression of nanos in the interior. And F, polycomb binding to lab, LAB, and DFD regulatory regions of embryo B caused the expression pattern. 3 minutes to solve this question. After 3 minutes, I'll switch the screen. Read all the options and try to correlate it with the graph which is given to you. 2 more minutes left. One more minute left. Okay, now I'm switching the screen. Hmm. Hmm. So Ajay has answered A and B. A and B. Okay. Hmm. Nice. And anybody else? Camelia, can you solve this question? Hmm? Were you able to understand this question or not? Tell me this first. Were you able to understand this question? No response? Why? No, ma'am. Okay, okay. You couldn't understand the question. Fine. Aditya, could you understand this question? Were you able to understand this question or not? Okay, fine. I'll explain. The first we'll try to understand the question and then we'll see how to answer this. Okay. So the question says, given below are the two graphs depicting gene expression in the anterior most area of the Drosophila embryo. So if this is your Drosophila embryo, this is the anterior region and this is the posterior region. So in the anterior region, the expression of two its genes. So there are two embryos. This is embryo A and this is embryo B. Okay, this is embryo B. So in embryo A, the expression of these two genes, LAB and DFD, is less in anterior region, whereas in the embryo B, you can see in embryo B, its expression is more. This LAB and DFD. Okay. And what are this? Can anybody tell me? LAB stand for labial and DFD is deformed. And these are segment polarity genes. We have just studied about them. See, these are expressed in the anterior regions. And this labial gene is responsible, and this deformed gene, it is responsible for the head region. Therefore, they are expressed in the anterior end. If this is the Drosophila act and this is the anterior region and this is the posterior region, this labial and deformed genes which are expressed in the anterior ends are responsible for the formation of the head region. Now the question says that we have two different embryos, and in these two different embryos, the expression of labial and deformed was measured, and it was found that the expression of labial and deformed in in embryo A was lesser in comparison to embryo B. That means in embryo B, they are expressed more in comparison to embryo A. Now tell me, now can you understand this question or not? Camelia, can you understand this question now? What does this question says? In the anterior region, your labial and deformed. These are the two segment polarity genes which are being expressed. And there are two different sets of embryos, embryo A and embryo B. And in both the embryo, the expression pattern is different. Now, based on our understanding that labial and deformed are required for the head region, for specifying the head region of the embryo, we have to see these questions now. Embryo A may have a bicoid deficiency, or and second is embryo A may have a hunchback deficiency. Now, can you tell me what is the function of bicoid and hunchback? So, bicoid and hunchback are required for specifying the anterior polarity. So, it might be the reason that because of bicoid and hunchback deficiency, the anterior region was not completely formed and therefore the expression levels of these particular genes was less. Can it be a case or not? Hmm? It can be a case. Why? Does it make sense? Okay. So, in this way, option A and option B both are correct. Because in A embryo, the two genes which are responsible for the head formation. So labial is important for the head identity, and deformed is also required for the head region only. Basically, it is needed for the mandibular and maxillary structures. So both labial and deformed, this is labial and deformed, they both are needed for specifying the head region. Now, since embryo A has a lesser expression of labial and deformed, it hints towards the fact that there can be a deficiency of bicoid and hunchback. Okay, so these two options are correct. Now, what is the next option? Embryo A may have an overexpression of nanos in the thoracic region. No, it cannot be correct. Why? Because nanos is responsible for the posterior development. So, if there is overexpression of nanos, it can inhibit the anterior gene expression bicoid, but the thoracic region will not directly be the result of the expression pattern which is observed here. So, even if there is an overexpression of nanos in the thoracic region, it doesn't matter because we are mostly concerned with the anterior most part. This is false. Then trithorax binding to labial and deformed regulatory regions of embryo cause the expression pattern. So, Triforex is another gene molecule which is involved for gene activation. So if trithorax bind, then there should be even higher expression of labial and DFT. But there is low. So this also doesn't go with our hypothesis. So if trithorax will bind to labial and deformed, there should be higher expression of labial and deformed because trithorax is involved in maintaining gene expression. Okay, so this is also wrong. Then embryo B may have had ectopic expression of nanos in the interior. So if nanos was ectopically expressed in the anterior region of embryo B, then the expression pattern of labial and deformed should be low. But here the expression level of deformed and labial in B, see here talking about B, in B is very high. So we cannot say that there is ectopic expression of nanos here. So this also becomes wrong. Then polycomb binding to labial and deformed regulatory regions of embryo B cause the expression pattern. Here again we come to embryo B. So basically polycomb group proteins are involved in gene repression. So if polycomb binds to labial and deformed, then the expression of labial and deformed should be less. But on the contrary, the expression of labial and deformed is very high in embryo. So this also becomes wrong. So always remember, trithorax is responsible for gene expression or gene activation, and polycomb is basically involved in gene repression. Okay, trithorax is involved in gene expression and polycomb is involved in gene repression. So if trithorax would have been expressed here, then the expression should be even more. And if polycomb would have expressed here, the expression should be less. So in this way, this question was solved. So is it clear or is there any problem? Camelia, could you understand the question, the options? Or is there any problem with any of the options? If there is any problem, you can tell me. See, see all the options and see the expression pattern. And if you have any problem in any of the options, let me know right now. See the expression patterns and the options. Okay, good. I hope this is clear to everybody. Okay, now we'll go to the next question. Hmm. The question is, misregulation in the expression of segment polarity genes may be a result of which of the following? Okay, so here we have segment polarity genes. So we have gap genes, then we have pair rule genes, and then we have segment polarity genes. And what are the examples of segment polarity genes? We have just studied. They are wingless, and then you have hedgehog proteins. These are all your segment polarity genes. Which, what does segment polarity genes do? They even give polarity to the segment. See, the name itself says segment polarity genes. That means it is providing polarity, the anterior, posterior polarity to each segments. Okay, so if there is any misregulation in the expression of the segment polarity genes, then what would happen? So your options are A, deletion of smoothen, B, overexpression of GS3K, C, loss of function mutation in wingless, D, loss of function mutation in hedgehog, E, loss of function mutation in pair rule genes, and F, loss of function mutation in biothorax. Okay, so you have 3 minutes to solve this question.
In this middle.
Yes, we never used to do that. I didn't even tell you. Like, because yeah, that's fine with me.
That should be away from this.
Yes, because last time this was
I told my about last time.
this was just
This is isopropyl alcohol. Understand?
oh, got it.
Keep that cassette. That one, that one. Ah, ah. Why you are dying?
Sir, because otherwise it catches fire, no? It will become black.
Should we catch?
No, but it becomes black, that's
No,
why.
no, no. Why? Why? Who knows? Who did that? It should be black.
It shouldn't be black.
No,
Oh, light shouldn't be black. So it becomes
just
black
you
if
have
I
to catch it. Wait. Give it from this side. Yes, go. Take this one. Huh?
yeah.
Okay, na?
Yeah. But see, it catches fire, sir.
Okay, no problem. What? Fire should be, it should catch fire. Don't do this like,
Oh,
That is, it is getting blacker. Over.
That's it.
Hmm.
Okay.
Close this, sir.
Yeah.
Yeah. Take this plate. Right on. Bring that plate. What name? Give it, give it. Come, come.
So could you solve this question? Hmm? Let me see. Okay, good. I think students have answered now. It's nice. Huh. So Ajay has answered all correct except F. Okay. Camelia has answered B, C, and D. And Aditya has answered C, D, and E. Okay. Okay. Just let me see this. Just a minute. Okay, we see this question. After this question, we'll quickly try to summarize whatever we have studied, because I think after this, uh, we have questions which are related to the plant. See, all the questions are plant related. So I was giving these questions, but just I wanted to know before starting the explanation. So, was plant portion also there in week 8? I don't think so. In week 8, plant portion was there. I think in week 9, lecture plant portions are there. Isn't it? Anybody
Answer means it is what it will go.
tell me?
And it is
So, was plant portion covered in week 8 or week 9? Hmm? You don't know? No response from anybody? Can anybody just see, go through your weekly lectures and let me know that whether plant portion was there in week 8 lecture or not? It is in ninth week, yeah. So I think these assignments are arranged in a way that we are getting questions of the upcoming portion also. Because in the last week also, the questions that we dealt in last week class, if you remember, the questions where bike white was injected in the middle and all. Those questions were actually taught in this week lecture, 8th week. I see this. So I think it's like this. It is 1 week ahead. So I won't take it that way, because then it will be difficult for you to relate everything. So plant questions we'll take in the next class when you will read about the plant thing. Okay, so first we'll try to complete everything regarding Drosophila this week, and by then next week when we will start the ninth week this thing lectures, then we'll do questions related to ninth week. Otherwise, even if I deal, it's difficult for you to manage it. Okay. So, anyways, first we'll try to solve this question, because this is an 8th week class. I mean, the content which were dealt in eighth okay, so 8th week, I expect you all that in 8th week class you study about the 8th week content. But since plant part was there in the 9th week content, so those questions will be dealt after you are after you have read the 9th week class, that is the next week. Could you understand what I'm trying to say? Okay. So, accepted answers for this question is A, B, C, D, and E. So, except F, everything is correct. So now see what you have answered. So, huh. So Ajay has answered all correct except F. Okay, good. And others have also answered correct only. Like you have chosen B, C, and D. B, C, and D. Okay. And Aditya has taken C, D, and E. C, D, and D. Okay. So now we'll see how A is also included and how other options are also included. So for this, let us again try to see this figure. So you just have to click. Just a minute, I'm sorry for this noise. Hmm. So I'll paste it here so that it becomes easier for you to understand. Okay. Now, see, it would be easier for you now. Now, see here, question is misregulation in the expression of segment polarity genes may be a result of which of the following? So, we studied that the expression of segment polarity gene is controlled via this cycle. And what happens in this cycle is that these are the wingless producing cells and these are the engrilled producing cells. So wingless producing cells, they transcribes the wingless and this wingless protein is secreted. And this secreted wingless protein, it goes and it binds to the Fizzle receptor. This Fizzle receptor is present in the engrilled producing cell. And through the whole process, through the downstream processing, transcription of engrilled as well as hedgehog take place, and this hedgehog is again a secretory molecule, secretory protein. It is secreted from the engrilled producing cell and it goes and it binds to the patch receptor. So this is the whole mechanism which uh, regulates the expression of segment polarity genes. So I'm writing here this mechanism regulates segment polarity genes. What does it mean? Any problem, any mutation, any deletion in this particular mechanism, in this cycle, will cause the misregulation in the segment polarity genes. Now, deletion of smoothened. So, if smoothened is deleted, then this patch receptor cannot bind to hedgehog because patch along with smoothened protein helps in the binding of hedgehog. If hedgehog is not bind, wingless transcription will not take place. If wingless transcription will not take place, it cannot bind to Fizzle proteins, and therefore transcription of engrilled and hedgehog will not take place. So that means if there is a deletion of smoothened protein, then there would be misregulation. So A is the correct answer. Now comes over expression of GS3K. Now, what is GS3K? GS3K is basically, if you remember the Wnt pathway, this GS3K is involved in, is associated in the beta destruction complex. If you remember the Wnt pathway, we have studied Wnt pathway in the last week. So this GS3 kinase is involved in beta destruction complex. So basically, it destroys the beta catenin. And if beta catenin is destroyed, then the transcription of engrilled and hedgehog cannot take place. Here, your armadillo is the base beta catenin, which when goes to the nucleus, it gives signals for the transcription of engrilled and hedgehog. That means because of the binding of your wingless protein, this beta destruction complex is destabilized and it is not able to bind with this beta catenin to destroy it. And therefore, this beta catenin can go inside the nucleus and do the transcription of engrailed and hedgehog. But if there is overexpression of this genus kinase, sorry, GSK3 kinase, then it can bind to beta catenin and destroy it. And if there is destruction of beta catenin, then there would be no transcription of engrailed and hedgehog. And when there would be no transcription of engrailed and hedgehog, then again the whole mechanism will get disturbed. So B is also the correct option. That is overexpression of GS3K. Then loss of function mutation in wingless. So if what is the function of wingless protein? To bind to the Frizzle receptor and the whole cycle take place. If there is a mutation in wingless protein in such a way that its function is lost, then the whole cycle will not take place. And if this whole cycle will not take place, then there would be misregulation in the segment polarity gene. Then loss of function in hedgehog. Where is hedgehog? Here. So if there is a mutation in the hedgehog protein, what will take place? Loss of function means mutation has led a hedgehog in such a way that it has lost its function. So now hedgehog will not go and bind to here. If hedgehog will not go and bind to here, again the cycle is disturbed. So this will also lead to misregulation. Loss of function mutation in pair rule genes. Now, can anybody explain why loss of function mutation in pair rule genes can lead to the misregulation of segment polarity gene expression? Because many students have answered E. I want to listen it from you. Why you think a loss of function mutation in the pair rule gene can misregulate the expression of segment polarity genes? Anybody? Hmm? What do you think? What will happen? We are talking about segment polarity genes, no? So how does pair rule genes will affect anything related to segment polarity genes? Ajay, can you give me the answer of this question? I am asking how the loss of function mutation in pair rule genes affect the expression of segment polarity genes. Think. Hmm? Hmm. So Aditya has very correctly said that pair rule genes regulate the segment polarity genes. Very good, Aditya. So we have already studied just now or in before that the genes, gap genes, pair rule genes, segment polarity genes, they work in a hierarchical fashion. Okay? Hierarchical fashion means that gap genes regulate pair rule genes, pair rule genes regulate the expression of segment polarity genes, and segment polarity genes and pair rule genes in combination affect the expression of homeotic genes. So there is a hierarchical regulation of these genes. So if there is any misregulation in the expression of segment pair rule genes, that means that all the genes, all the type of genes which are downstream of pair rule genes will be affected. So what is the immediate downstream genes of pair rule genes? They are the segment polarity genes. So if there is any loss of function mutation in the pair rule genes, then the expression of segment polarity gene is also inhibited. So this is also correct. Sorry. Then last option is loss of function mutation in bithorax complex. So if there is any loss of function mutation in bithorax complex, will segment polarity genes be affected? Yes or no? If yes, why? If no, why? You think if there is any loss of function mutation in bithorax, will segment polarity genes be affected or not? What are bithorax genes? First, tell me this. What are bithorax genes? Under which category do they belong? Bithorax. Hmm? So,
Yeah.
bithorax genes.
Hmm.
Under which category do they belong? These are gap genes or pair rule genes or segment polarity genes or homeotic selected genes. Bithorax are nobody knows. Aditya, Ajay, homeotic genes. Correct. So, bithorax genes are the homeotic genes. That means what? That means these genes are present downstream of the segment polarity genes. So, after segment polarity genes give polarity to each segments, then your homeotic genes come into function. Then they give the identity to each segments. That means that even if there is a loss of function mutation in the bithorax complex, I mean, even if there is a loss of function mutation in the homeotic genes, there would be no effect on the segment polarity genes because it is after it. So F would be wrong. Okay. So could you understand this question, Camilla? Is it clear to you? Could you understand? Good. So from this we can say, just a minute. First we have maternal genes, which are, this is the crux of the whole Drosophila axis development that we have studied. Bicoid, caudal and hunchback, and nanos. It is before fertilization. These are before fertilization. After fertilization, we have zygotic genes. And all the zygotic genes except your hunchback, are, I mean, all the gap genes except your hunchback are the zygotic genes. Hunchback is both maternal gene as well as zygotic gene. So you have gap genes, okay, and gap genes, it gives your, okay, what I'll do is I'll make a flow chart. So gap genes, it gives you a broad regional definition. After gap genes comes your pair rule genes. Pair, and these pair rule genes are regulated by gap genes. That means gap genes regulate the expression of pair rule genes. It divides the embryo into what? Into stripes. And these stripes are horizontal or vertical? These are vertical stripes. These pair rule genes regulate the expression of segment polarity genes. And these genes, they establish polarity to each segment. That means each segment now gets a anterior and posterior axis kind of polarity. So they establish polarity to each segment and they refine the segment boundaries. That means, see, this is a very important slide, I would say. That means there were first gap genes made some broad divisions. Okay. After broad divisions, pair rule genes made even more refined division. Now the divisions are even more refined, and now our divisions are completed. Now once the divisions are completed, these segment polarity genes will, along with the pair rule genes, they will help in the expression of homeotic genes. And these homeotic genes, it gives identity to each segment. And identity in what form? Identity, that means whether that particular segment will be converted into a head or thorax or abdomen. So this is the hierarchical classification. So that is why we said that loss of function mutation in bithorax will not affect segment polarity genes, because bithorax are the homeotic genes. Okay. If you see this figure, uh, it is here. See here, there are two types of homeotic genes. First is antennapedia complex, and second is your bithorax complex. So your homeotic selective genes also have two complexes: antennapedia complex and bithorax complex. Antennapedia complex have these four genes, and bithorax have these three genes. So bithorax is a complex of segment polarity genes. So just a minute, I'll delete these slides because it has nothing in it. Does this slide has anything? Sure. So I hope this question is clear. So is this question clear to everybody? And more importantly, the hierarchical chain network that helps in the development of Drosophila body axis. Of course, there are many other genes which are involved. These are not the only genes which are involved. Okay, there are many other genes as well. But these genes are this general classification is important. And rest, if you want to study more about this, you can go and read Gilbert's for developmental biology. Is best book for this subject. And the recordings of this is my name. You can go to the YouTube channel and you can get it. So is it clear to everybody? Is there any question? Camellia? No. Okay. Then we'll end the class here. Bye, and we'll meet in the next week. Have a great week, and we'll meet in the next week. I think for next week we'll meet on Tuesday. Sorry, Monday. For this week I kept it on Tuesday, but next week we'll meet on Monday. Okay. Bye, everybody.
Yeah, ma'am, thank you.
Okay, bye. So I'm just marking the important points and I'm deleting the slides which are not important so that anybody who goes through the video can understand it. For the record, the questions, we try to solve all the questions, and the questions which are left, we always complete them in the next class. This is for the record. Thank you so much.